Hello guys, how have you been doing? I hope you're doing well, enjoy your home, yeah? So today I'm gonna talk about meiosis. This is a continuation about cell division. So if you didn't watch the mitosis video before, I highly recommend you watch because this is a continuation and you're gonna be lost if you don't understand mitosis first, okay? So before I start talking about meiosis, I'm going to do a comparison between mitosis and meiosis. Both of them are cell division, but both of them are cell division, but they have different purpose. For mitosis, as you remember, we have one mother cell which is 2N, resulting two daughter cells that are also 2N. What does that mean? This means that the number of chromosomes from the mother cell, is the same in the daughter cells. Okay, this is going to be different from meiosis. So, mitosis. Each mother cell divides and forms two somatic. So, what's somatic? Somatic is the cells of your body. Any cell of your body except the sexual, your gametes, okay? And mitosis forms two somatic cells. Each cell that divides forms two somatic cells that are identical to the mother cell or to the parent cells. So we call them diploid cells because they keep the same number of chromosomes. So in summary, mitosis forms somatic cells, cells of our body, and each mother cells produce two daughter cells with exactly the same number of chromosomes. So we keep the same number of chromosomes. For example, for human, we have 46 chromosomes, and each cells, when it divides, also have 46 chromosomes. Okay, so this is a summary of mitosis. Now, what's the difference between mitosis and meiosis? For meiosis, we also start with a 2N cell or a diploid cell, but I have two divisions. So I have meiosis 1. This number of chromosomes reduced by half. So instead of have 2N, now I have two cells of 1N. So in human, I had 46 before. Now I have two daughter cells that are 23 each. So I reduce by half. And after that, I have another cell division that we call meiosis 2, but the number of chromosomes keeps the same. So I formed 4 new cells that are 1N or half number of chromosomes. This is the main difference between mitosis and meiosis. So meiosis, its cells divide and form 4. For mitosis, it was 2. And for what kind of cell? Germ cells or your gametes, with one copy of each chromosome. So we call them haploid cells because they have just half of the number of chromosomes. So just to remember, somatic cells are cells of your body. So all cells of your body we call somatic, except germ cells. So germ cells is the reproduction cells or your gametes. That's the only cells that are not somatic. And how do I form the germ cells? By meiosis, okay? And all the somatic cells by mitosis. So this is the main difference. Now I'm gonna focus more on meiosis, okay? So meiosis is the process of cell division resulting haploid cells or gametes. How does this happen? So we have one mother cell. In this case, I have just one pair of chromosome or two chromosomes, and we call them 2N. Okay, 2N means that for each two chromosomes, I have one pair. Okay, so in this case, my 2N is equals to 1 because I have just one pair. Before I start any cell division, meiosis or mitosis, I need to duplicate. Remember when I duplicate? In the interphase. So I duplicate in the interface. What happens when I duplicate the cell? I duplicate the number of chromosomes. So here I have 2 chromosomes, but now each copy of the chromosomes I call chromatid. Okay, so when I duplicate the cell, I call them chromatids. So each leg here, so each part here of the chromosome is one chromatid. So in this case, I have 1, 2, 3, 4 chromatids, but I still have just 1 and 2 chromosomes. The only difference is that I have a copy of each chromosome, okay, but my genetic material is the same. And now I call it 4n because I duplicate the number of chromosomes that I have. So in meiosis 1, what happens? I told you before. The number of chromosomes reduced by half. So what's going to happen to my cell here? I'm going to split the cell in 2. So each chromosome goes to one cell. So now I have just one chromosome duplicated in each cell. Okay, so I have one chromosome, but I have 2 chromatids because I have one copy of each chromosome. And this one also one chromosome with 2 chromatids. And in this case, I had 4n before. If I reduce by half, I'm going to have 2n. So now I have 2 cells of 2n. After that is not finished, I need to perform the second meiosis. Okay, so I do meiosis 2. What happens in meiosis 2? Each of these chromat that are held by a centromere, each of them are going to be separated. So I have these 2 chromosomes here, these 2 chromatids, sorry, they split in 2. So I'm going to split them in 2, and each of them are going to one cell. Now I have just n number of chromosomes, not 2n anymore. So I have in each cell just one chromosome. Why I don't call chromatid now? Because it's not duplicated. I just call chromatid when I have a copy of each chromosome, okay? So it's just terminology. In this case that I have just chromosome, I have just one copy, so I don't call them chromatids anymore. I call them just chromosome. So in summary, meiosis. One mother cell, for example, with 2 chromosomes or one pair. First, ela duplicate, so in the interface, they duplicated, and I have now 2 chromosomes, but duplicate, so I have now 4 chromatids. Why I call chromatid? Because I duplicate, I have a copy of each. Now I go to first meiosis. What happened? I split these 2 chromosomes in 2, so I reduce my number of chromosomes by half. So half of my chromosome goes to one cell, and the other half goes to the other cell. After that, I perform meiosis 2, and I split the chromatid. The copy of each chromosome goes to one cell. Now I formed 4 daughter cells with half number of the chromosome of my mother cell that I had before. Why this is important? Remember, they form gametes. Why it's important to have half? Because when I make a baby, I need to keep the same number of chromosomes. If my gametes has, for example, 46 chromosomes, my baby is going to be 92, and his baby is going to be 184, and so on. It doesn't happen like that. So I need to reduce my number of gametes in half every time. So when I make one baby, so it receives one copy of the chromosome from the daddy and one copy from the mommy. Now it's gonna keep the same number of chromosomes over and over in this piece. Okay, I'm gonna do the same process, but now let's count together the number of chromosomes in human cells. So the draw is gonna be different. So the picture is the same, but now let's count the number of chromosomes of the human cells. So one mother cell have 23 pairs or 46 chromosomes. What happens first? What needs to happen first? So here it's 2n. What's going to happen? Duplication. So what's going to happen? Duplicate of each chromosome. So now I call them chromatids. I still have 46 chromosomes, but duplicated. So I have 92 copies, or each copy I call chromatids. And now I have a cell that's Rn. Why? It's duplicate the number of chromosomes. What happened? What's the first meiosis that I have? Meiosis 1. What's going to happen here? My chromosomes will split in two. So imagine I have 46 chromosomes. Now I'm going to split. So half of the chromosomes go to one cell, half to the other cell. So if I had 46, now I have 23. And my chromatids as well. So I had 92, now I have 46. I reduced by half. So each cell is reduced by half. I have 23 in one side, or 46 chromatids, and the same for the other one. And now I have two daughter cells that are also 2n, but it's not ended. Why? Because I have 23 chromosomes, but they are duplicated. I have two copies of each. I don't want two copies of each. I want to reduce this by half. So I'm going to separate my chromatids. And in meiosis 2, I have two cells. So I separate each chromatid. And now I formed 4 cells that has the half number of chromatids. So each cell is going to be 23 chromosomes. I don't call them chromatids anymore because I don't have a copy. Remember, I just call chromatid when it's copy. Now I have just one copy of each chromosome. So I have 23 chromosomes in each cell, not 23 pairs. Look at the difference between the mother cell. So look here in the mother cell. I have one pair. My daughter cell, just one. Okay, so when I make a baby, I can give them one chromosome that I received from my daddy, or one chromosome that I received from my mom. And this happens with all the 23 chromosomes that we have. Okay, so it's impossible for me to give my child all the chromosomes I have. I just give them half. And what's going to be this half? That is randomly. We never know. But could be, for example, I could give all the chromosomes that I received from my daddy. It can happen, okay? Or all that I received from my mom, or half, or more of my mom, more of my daddy. So it depends, okay? So that's it for meiosis, for the counting. Now let's see the phases, okay? So just to remember, what's haploid cell? Haploid means simple. I have simple number of chromosomes, or half, okay? This is just happening in gametes. So as we told before, meiosis has two phases, meiosis 1 and meiosis 2. But that's not enough, you need to know more, okay? So meiosis 1, what does? Reduce the number of chromosomes by half. And remember the 4 phases of mitosis? What were the 4 phases for mitosis? Prophase, Metaphase, Anaphase, and Telophase. Good. Now for meiosis, I also have the same phases, but 2 times. Okay, the names are the same, but I have 2 times. So for meiosis 1, I have Prophase 1, Metaphase 1, Anaphase 1, and Telophase 1. And what's going to happen for meiosis 2? I also have Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. Okay? And if you know the phase, what happens in each phase for mitosis, you know almost everything for meiosis. Okay? The difference is I have 2 of each for meiosis, so meiosis 1 and meiosis 2. What's the difference between them? Meiosis 1 reduces the number of chromosomes by half. So I split my chromosomes for each cell, daughter cell. And the meiosis II, what I separate each chromatid, or I separate each chromosome into 2 strands. So each chromatid of each chromosome goes to one side. Now let's go in detail what's gonna happen. So in prophase, who remembers what's prophase in mitosis? Prophase, what happens? Chromosome condensation. So this is the key, is the same. The chromosomes will condensate. Remember the name of the protein that helps the chromosome to condensate? Yes, it is histone. Very good. And what else happens? My nuclear membrane dissolves, so disappeared my nuclear membrane. What also happens? Formation of a spindle. This spindle here, so this part here, what it does, it helps the chromosomes to go to the middle of the cell, okay? Now, it also happens crossing over. Crossing over is a kind of genetic variation. This is really important, but I'm gonna talk about this later, okay? First, I want you to understand the meiosis. Then I go over and talk about crossing over. So hold on a second. Okay, I'm not gonna explain for you right now because it's a lot of information. But in the next video, I'm gonna explain what's crossing over, okay? So after prophase, what's the next phase? Metaphase. Meta, remember, M stands for middle. So the chromosomes goes to the middle, and the spindle helps the chromosomes stay in the middle. Now, what happens? In mitosis, the chromosomes align in just one line. Remember the animation that I did for you? But now I have the chromosomes homologous, so the chromosomes that I received from daddy and my mom, they go to the middle together. So I call them tetrad. Okay, they go to the middle together. It's not just it's a line, but it's like a double line. Okay, that's the main difference between meiosis and mitosis. So the homologous chromosome, what's homologous chromosome? Chromosomes that contain the same kind of genes. Okay, for example, genes for color of eyes. I receive one gene from my daddy and one gene from my mom. They are not exactly the same, but they do the same job, that the color of my eyes. So here, the homologous chromosomes go to the center of the cell. Okay, so they go to the center of the cell. After metaphase, what's the next phase? Remember, Anaphase. What's Ana again? Separation. Now what do I separate? I'm not separating chromatid yet. I'm separating the homologous chromosome. So each chromosomes that were together before in the middle, they go to the opposite poles of the cell. So they goes to the opposite poles of the cell. So half of the chromosomes go to one side and the other half goes to the other side. This happens in anaphase. Anaphase always is separation. In this case, I'm separating the homologous chromosome. And the last one is telophase. So here I'm not distinguishing cytokinesis, but you know that cytokinesis is just the end, okay? So telophase, what's gonna happen? Now I'm gonna form again the nuclear membrane and the cells just divide. That's it. So I form two cells that have half of the chromosomes. So half of the chromosomes here, half of the chromosomes over there. This is the meiosis 1. It's not over. Meiosis has more. Meiosis 2. What happens again? Prophase 2. What do you think is going to happen in prophase 2? Condensation again. Okay. Nuclear membrane dissolves again. And formation of a new spindle. Okay. So this is spindle here. My centriolus does this spindle. After prophase 2, what's going to be? Metaphase 2. Meta, middle. What's going to the middle now? Now I don't have the tetrad because I separate my chromosomes homologous. So my chromosomes goes and do a single line, just like in mitosis. Okay? The only difference, now they are separate already, so they form a central line. They go to the central line alone, so now it's single line. So chromosomes line at the equator of the cell, so goes to the middle. After anaphase, what ana does separate. So ana always separate. Remember this. So what's going to separate now? Do I have chromosome homologue here? Homologous chromosome? No, I have chromatid. Remember, chromatid is a copy. Now I'm separating the copy. So the centromere is the center that holds the chromatid together. They separate, they divide, and each chromatid moves to the opposite side of the cell. So each chromatid goes to one side of the cell. And finally, what's the last one? Telophase. Okay, what's going to happen in telophase? Nuclear membrane again, always the same. So nuclear membrane and the cytokinesis in the end of telophase. Okay, and all the chromatids that were separate goes to one cell. Oh, how many cells are formed here? 1, 2, 3, 4 cells, right? In mitosis, I just formed 2, remember? In meiosis, I formed 4. So that's it. Take a breath and let's do a summary. Let's compare now mitosis and meiosis. I recommend you to pause this video and try to complete by yourself, okay? So, for example, number of stages. How many number of stages mitosis I have? How many phases? How many, for example, prophase, telophase, how many? Mitosis, how many I have? 4. What are they? Prophase, metaphase, anaphase, and telophase. I just have 4. For meiosis, how many do I have? 8. Why 8? Because I have 2 meiosis, as if I had, for example, mitosis 2 times. So I have meiosis 1 with prophase 1, prophase metaphase 1, anaphase 1, and telophase 1. But it's not over. What do I have next? Meiosis 2. What I have in meiosis 2? Prophase 2, metaphase 2, anaphase 2, and telophase 2. So I have 8. I have double. Now, what type of cells are formed in mitosis and meiosis? Somatic or germ? Mitosis. Mitosis I have somatic. What somatic cells again? The cells of our body. Good. And meiosis, what cells I form? Germ cells or gametes. That's a huge difference between them. Next one, what's the purpose? Why do I perform mitosis? Why do I need to form somatic cells? Why? For what? For growth, for cell proliferation, for me to grow or for me to heal. For example, when I have a cut, I need to replace the cells. So for cellular proliferation, to replace the cells of your skin, for example, okay? And meiosis, what's the purpose? Why do I do meiosis? Why this is important? To what? For sexual reproduction. Yes, if I don't do meiosis, I don't form gametes and I don't reproduce sexually. Okay, and this is really important to preserve a species. And how many cells per each mother cell I form? So if I have just one mother cell, how many cells are formed in mitosis? 2 daughter cells. And for meiosis, how many? 4 daughter cells. Very good. So mitosis forms just 2 for each and meiosis 4, but they are different. How many number of chromosomes do I have for each mitosis? So remain the same. In human species, I have 46, so remain the same number. And for meiosis, I reduce by half. So I have just 23 in each daughter cell. And finally, what the type of cell formed? I mean, they are diploid, haploid, tetraploid. Which type of cells mitosis does? Diploid cell. Very good. And meiosis? Haploid. So why haploid? Because I have half number of chromosomes. Okay, guys, I guess that's it. I know it's a lot, but you can watch this video over and over as many times as you need, okay? And please watch this very carefully and answer the questions in the form for you to get some practice, okay? And if you have any question, please contact me, okay? Thank you so much and have a wonderful day.